Hello, my name is Aisha Qureshi, and today I will be telling you about my project on the effect high concentration lachrymatory chemicals have on redworm sensory neural synapse. The ultimate goal of this research is to illustrate that lachrymatory agents have detrimental effects on the sensory neuron of redworms when they have exposure. By using lachrymatory agents like nonavamide, onion, and pepper spray, this illustration can prove that tear gas, a detrimental lachrymatory agent that harmfully stimulates sensory neurons, has detrimental effects on a human's peripheral nervous system after exposure. The hypothesis of my project was, if a sensory neuron is exposed to a high concentration lachrymatory agent, then it will have a faster electrical transmission because the lachrymatory agent will excite the neuron, thus causing nerve damage because of the fast pathway that is induced through the pain that occurs when the worm is in contact with the chemical. A sensory neuron is a nerve cell that transmits sensory information from the peripheral nervous system to the central nervous system, like sight, smell, taste, touch, and sound. Tear gas is a chemical weapon that activates corneal nerves in the eyes, causing extreme irritation and pain. It irritates mucous membranes in the eyes, nose, mouth, and lungs. Thereof, it is a lachrymatory agent. Synapse facilitates the transfer of information in a neuron. This transfer is susceptible to chemicals, increasing or decreasing the speed of the transfer. A nociceptor is a receptor of a sensory neuron that responds to potentially damaging stimuli by sending signals, also known as synapses. An axon is a long thread-like fiber of a neuron which carries outgoing messages from the neuron's cell body. A dendrite, on the other hand, is the fiber of a neuron that brings information to the cell body. In this experiment, axodendritic synapse is apparent. It is a synaptic contact between an axon of one nerve cell to the dendrite of another neuron. Figure 6 right here illustrates the mean conduction velocities of the lacrimal chemicals in comparison to each other and their controls. A two-sample t-test comparing each sample to its control indicated that every sample is significant at the 5% significance level. However, there must be caution taken when understanding this because of the very low sample sizes. Finally, the nonavamide sample was compared to the water control because the 100% ethyl alcohol sample had too many voided trials due to excess static that disrupted the wave output. So, in conclusion, chemicals that have lachrymatory agents affect the speed of a neuron's synapse. These chemicals have a lasting effect on the neuron long after the time of contact. You can see the effect the chemicals have on the worm a day after while also seeing it mathematically. Low concentration lachrymatory agents have a higher effect on the sensory neuron. And finally, high concentration lachrymatory agents are probably too strong for the neuron, maybe killing it little by little, and so it results in a lower conduction velocity. This research is really important because tear gas is a lachrymatory agent, which, as a conclusion from this experiment, detrimentally affects the sensory neurons of the human body. In addition, tear gas affects the peripheral nervous system of a human, actually hurting the human long after time of contact. Finally, this research shed light on the necessity of restricting tear gas use by state and local police forces.